Diffusion is the movement of molecules from a higher concentration to a lower concentration, that is, down their concentration gradient, until equilibrium is achieved and they are distributed equally. Diffusion is a physical process due to random molecular motion that can be observed with any type of molecule. For example, when a crystal of dye is placed in water, the dye and water molecules move in various directions, but their net movement, which is the sum of their motion, is toward the region of lower concentration. Eventually, the dye is dissolved in the water, resulting in equilibrium and a colored solution. A solution contains both a solute, usually a solid, and a solvent, usually a liquid. In this case, the solute is the dye and the solvent is the water molecules. Once the solute and solvent are equally distributed, they continue to move about, but there is no net movement of either one in any direction. The chemical and physical properties of the plasma membrane allow only a few types of molecules to enter and exit a cell simply by diffusion. Gases can diffuse through the lipid bilayer. This is the mechanism by which oxygen enters cells and carbon dioxide exits cells. Also, consider the movement of oxygen from the
Elaborate.
of the lungs to the blood of the lung capillaries. After inhalation, the concentration of oxygen in the alveoli is higher than that in the blood. Therefore, oxygen diffuses into the blood. Several factors influence the rate of diffusion. Among these factors are temperature, pressure, electrical currents, and molecular size. For example, as temperature increases, the rate of diffusion increases. The movement of fish in the tank would certainly speed the rate of diffusion. The diffusion of water across a differentially permeable membrane due to the concentration differences is called osmosis. To illustrate osmosis, a thistle tube containing a 10% solute solution is covered at one end by a differentially permeable membrane and then placed in a beaker containing a 5% solute solution. The beaker has a higher concentration of water molecules, lower percentage of solute, and the thistle tube has a lower concentration of water molecules, higher percent of solute. Diffusion always occurs from higher to lower concentration. Therefore, a net movement of water takes place across the membrane from the beaker to the inside of the thistle tube. The solute does not diffuse out of the thistle tube. Why not? Because the membrane is not permeable to the solute. As water enters and the solute does not exist, the level of the solution within the thistle tube rises. In the end, the concentration of the solution in the thistle tube is less than 10%. Why? Because there is now less solute per unit volume, and the concentration of solute in the beaker is greater than 5%. Why? Because there is now more solute per unit volume. Water enters the thistle tube due to the osmotic pressure of the solution within the thistle tube. Osmotic pressure is the pressure that develops in a system due to osmosis. In other words, the greater the possible osmotic pressure, the more likely it is that water will diffuse in that direction. Due to osmotic pressure, water is absorbed by the kidneys and taken up by the capillaries in the tissues. Osmosis also occurs across the plasma membrane, as we shall now see in figure 5.8. Isotonic solution. In the laboratory, cells are normally placed in isotonic solutions, that is, the solute concentration and the water concentration, both inside and outside the cell, are equal, and therefore there is no net gain or loss of water. The prefix iso means the same as, and the term tonicity refers to the strength of the solution. At 0.9% solution of the salt sodium chloride, NaCl, is known to be isotonic to red blood cells. Therefore, intravenous solutions medically administered usually have this tonicity. Terrestrial animals can usually take in their water or salt as needed to maintain the tonicity of their internal environment. Many animals living in estuaries, such as oysters, blue crabs, and some fish, are able to cope with the changes in the salinity, salt concentrations of their environment. Their kidneys, gills, and other structures help them to do this. Hypotonic solution. Solutions that cause cells to swell or even to burst due to an intake of water are said to be hypotonic solutions. The prefix hypo means less than and refers to a solution with a lower concentration of solute, higher concentration of water than inside the cell. If a cell is placed in a hypotonic solution, water enters the cell. The net movement of water is from the outside to the inside of the cell. Any concentration of a salt solution lower than 0.9% is hypotonic to red blood cells. Animal cells placed in such a solution expand and sometimes burst due to the buildup of pressure. The term cytolysis is used to refer to disrupted cells. Hemolysis, then, is disrupted red blood cells. The swelling of a plant cell in a hypotonic solution creates turgor pressure. When a plant cell is placed in a hypotonic solution, we observe expansion of the cytoplasm because the large central vacuole gains water and the plasma membrane pushes against the rigid cell wall. The plant cell does not burst because the cell wall does not give way. Turgor pressure in plant cells is extremely important to the maintenance of the plant's erect position. If you forget to water your plants, they will due to decreased turgor pressure. Organisms that live in fresh water have to prevent the uptake of too much water. Many protozoans, such as Paramecia, have contractile vacuoles that rid the body of excess water. Freshwater fishes have well-developed kidneys that excrete a large volume of dilute urine. Even so, they have to take in salts at their gills. Even though freshwater fish are good osmoregulators, they would not be able to survive in either distilled water or a marine environment. Hypertonic solutions that cause cells to shrink or shrivel due to loss of water are said to be hypertonic solutions. The prefix hyper means more than and refers to a solution with a higher percentage of a solute, lower concentration of water, than the cell. If a cell is placed in a hypertonic solution, water leaves the cell. The net movement of water is from the inside to the outside of the cell. Any concentration of a salt solution higher than 0.9% is hypertonic to red blood cells. If animal cells are placed in the solution, they shrink. The term crenation refers to red blood cells in this condition. Meats are sometimes preserved by salting them. The bacteria are not killed by the salt, but by the lack of water in the meat. When a plant cell is placed in a hypertonic solution, the plasma membrane pulls away from the cell wall as the large central vacuole loses water. This is an example of plasmolysis, a shrinking of the cytoplasm due to osmosis. The dead plants you may see along a salted roadside died because they were exposed to a hypertonic solution during the winter. Also, when salt water evades coastal marshes due to storms and human activities, coastal plants die. Without roots to hold the soil, it washes into the sea, doing away with many areas of valuable wetland. Marine animals cope with the hypertonic environment in various ways that prevent them from losing water to the environment. Sharks increase or decrease urea in their blood until their blood is isotonic with the environment, and in this way do not lose excess water. Marine fish and other types of animals drink no water but excrete salt across their gills. Have you ever seen a marine turtle cry? It is ridding its body of salt by means of glands near the eye.
It's so sad.
Facilitated transport. The plasma membrane impedes the passage of all but a few substances. Yet, biologically useful molecules are able to enter and exit the cell at a rapid rate, either by way of a channel protein or because of carrier proteins in the membrane. These transport proteins are specific. Each can transport with only a certain type of molecule or ion, which is then transported through the membrane. It is not completely understood how carrier proteins function, but after a carrier combines with a molecule, the carrier is believed to undergo a change in shape that moves the molecule across the membrane. Carrier proteins are utilized for both facilitated transport and active transport. See Table 5.1. Facilitated transport explains the rapid passage of water and across such molecules as glucose and amino acids across the plasma membrane. Whereas water moves through a channel protein, the passage of glucose and amino acids is facilitated by their reversible combination with carrier proteins, which transport them through the plasma membrane. These carrier proteins are specific. For example, various sugar molecules of identical size might be present inside or outside the cell, but glucose can cross the membrane hundreds of times faster than other sugars. As stated earlier, this is the reason the membrane can be called differentially permeable. A model for facilitated transport, figure 5.9, shows that after a carrier has assisted the movement of a molecule to the other side of the membrane, it is free to assist the passage of other similar molecules. Neither diffusion nor facilitated transport requires an expenditure of energy because the molecules are moving down their concentration gradient in the same direction they would tend to move anyway.
The plasma membrane allows only certain molecules to pass through it. Some molecules, such as water and gases, simply diffuse across the membrane from higher to lower concentration. This process doesn't need energy to happen.